All right, this is the Spanish River Biology Review Part 1. What we're going to do is we're going to go over the biology review paper that you guys got in class. We're going to go over it in 3 sections, 3 25-minute sections, and it'll be everything you need to know for the exam. All right, so here we go. First thing asked you was list and describe the steps in the scientific method. First step in the scientific method is you have to identify a problem. Second thing you're going to do is create a hypothesis. That's an educated guess. Third thing you're going to do is you're actually going to conduct an experiment like you did in class. You're doing experimentation, collect data. The last step is you draw a conclusion based on analyzing the data, see whether your hypothesis is correct or incorrect. So those are the key steps in the scientific method. You just have to be able to identify the general steps. That's it. Why do many experiments make use of a control group? A control group is the standard to compare to the experimental group. That's the key part of it. So what you're doing is you're using a control as a standard for comparison. So if you're testing plants, acid rain, one set of plants only gets water, normal conditions, and then you test another set with acid rain, and you can compare what happens to the two experimental groups. So a control is just a standard for comparison. What's the difference between a hypothesis, a theory, and a law? Well, you should know a hypothesis is an educated guess. A theory is a possible explanation for something that you can test and test, but really never gets proven. It's just a possible explanation because it's impossible to disprove it. Scientific law is something that the key thing is observed. So it's something that you're observing, like the law of gravity. You can drop a book and it falls to the ground. So it's observable. So that's the difference between key thing you need to know is a hypothesis is an educated guess. Theory is a possible explanation, and a scientific law just means that it's actually been observed. Next one, difference between an independent and a dependent variable. Independent variable is the variable that you change during a experiment. So during a biology experiment, if you're watering plants with different amounts of fertilizer in them, and you vary changing the amounts of fertilizer, 10 grams, 15 grams, 20 grams, that's the independent variable. It's the variable that you change during the experiment. Okay? Dependent variable is what you measure. So let's say you're watering these plants and they're putting different fertilizers in them, and they have different plant heights. So measuring the plant height is what you measure. So dependent variable is what's measured. The independent variable is what's changed during an experiment. What makes things organic? Anything that contains carbon can be considered organic molecule. So in order to be an organic molecule, you have to contain carbon. That's right on the exam, easy point right there. Compare and contrast the structure or function of the following organic molecules. Well, you know carbohydrates, what they are. They're in different foods. Two things that they do, they're short-term energy storage and a quick release of energy. Those are the two key components that carbohydrates do. The subunits, we're talking about sugars, monosaccharides, but really sugars is all you really need to know. Proteins. Proteins regulate cell process. Enzymes are proteins, and they're the basic building block of bodies and cells. So they give bodies structure. They regulate cell processes. The subunit is amino acids. So amino acids are what make up the organic molecules, macromolecules, or large molecules. Lipids, or fats, the key component in cell membrane, plus their long-term energy storage. Those are the two key things you need to know. Lipids are made up of fatty acids. Nucleic acids are the last group. They store and transmit genetic information. Their key little subcomponent, or the little piece that they're made out of, subunit, are nucleotides. Those are the 4 macromolecules that you learn in the first semester, and there's 1 or 2 questions on each. Really, you just need to know what the subunits are, and what the functions are. What does the term membrane-bound organelle means? It means that the organelle is separated from the cytoplasm, so there's a barrier from the cytoplasm. That's all that means. What type of cells are they found in? They're found in eukaryotes. So eukaryotic organisms. What are the 3 parts of cell theory? First part is all cells come from pre-existing cells. That's the first part of cell theory. All living things are made of cells, and the cell is the basic unit of life. Those are the three key parts of cell theory. What is the function of a cell membrane? It basically allows materials in and out of a cell. That's the function of a cell membrane. It allows materials to go in and out. Here's the membrane structure, fluid mosaic model. It allows certain molecules to go in, certain molecules to go out. In order of smallest to largest, the smallest unit is going to be a cell. So we have cell, net, which cells, you group a bunch of cells together, you get tissues, group a bunch of tissues together, and you get an organ. Couple organs together, you get an organ system. And that's the basic unit or basic levels of organization in biology. Next one, fill out the chart, give the letter as seen in the diagrams below. So, well, the function of the nucleus, control center for the cell. So it's the control center for the cell, controls all the activities. Type of cell it's in eukaryotic cells. So in eukaryotes, they're present. Function of the plasma membrane. Plasma membrane allows things in and out of cells. Allows in, out, materials in and out of the cell. They're in all types of cells. So prokaryote and eukaryotic cells. Cell wall. Function of the cell wall is it provides plant structure. Structure for plants. And support. Structure and support for plants. Cell walls are only in plant cells, nothing else. Mitochondria. Mitochondria is where cellular respiration happens in the cell, so it provides the cell with energy. Energy, because that's where ATP, the form of energy that cells can use, is formed. That happens in all eukaryotic cells. Vacuoles. Vacuoles are used for storage. So the whole point of having a vacuole is a storage, kind of like a cupboard in the kitchen, closet. You can store stuff in there. Cells store all kinds of stuff. They store water, other materials, nutrients. Vacuoles are also going to be in eukaryotes. Chloroplasts are where photosynthesis occurs. So photosynthesis occurs in chloroplasts. Those are only going to be in plant cells. Last but not least, ribosomes is where proteins, the whole point is to make proteins. So proteins are assembled there, and that's going to happen in all, basically it happens in all cells, really. So you have ribosomes, occurs in all cells. All right, which of the following cells is a plant cell? The one that's going to be the plant cell is going to be this one right here. You can tell by the shape of the cell. You can see the rigid cell wall right there. So the one on the left, you should be able to identify which one's a plant cell. Which structures are only found in the plant cell? Well, the first key thing is a cell wall. Only a cell wall is going to be found in a plant cell. Chloroplasts are only found in plant cells. The last thing is this little area right here, the central vacuole. Two key things you want to know are chloroplast and cell wall. So chloroplast is right here. You see that little structure right there on your little sheet. Corresponds to structure I is going to be the chloroplast. What structures are found only in the animal cell? Well, only in the animal cell. You have the centrioles right here. Centrioles and then lysosomes. Lysosomes are the two things that are only found in animal cells. All right, explain what has happened in the diagram. Remember, we talked about different types of diffusion in biology. Why did the dark molecules not move to the left? You see the dark molecules they're talking about right here? These are the dark molecules. They're too large. So they're too large. Remember, we talked about cell membranes are semi-permeable. Some molecules are too large to go from one side to the other. How is a semi-permeable membrane like a cell membrane? Well, remember, a cell membrane only lets certain things in and out of a cell. So it's the same idea. If it's a semi-permeable membrane, which you see right here, that membrane right there is semi-permeable. So only certain molecules that have the right size can travel through. If the dark molecule is starch, where is the starch concentration the greatest? Well, if you look, I have the dark molecules on the right-hand side and none on the left. So I'm going to say, whoops, that's not the pen I want to use. I'm going to say right, so the right side right there. If white molecule is water, where's the water concentration the greatest at first? Well, if you look here at first, you'll see on the left-hand side contains more water molecules. And then we undergo osmosis. Always goes from a concentration of high to a concentration of low. That's the definition of osmosis. Water, movement of water through a semi-permeable membrane. You have more water molecules on this side, so what you're going to see is they're going to move to that side. That's why the water level changes. If the dark molecules could move, they can't be there because they're too large, but if they could possibly move through the membrane, which direction would they move? They would move to the left. Why? Because they would go from a high concentration, which you notice is on the right-hand side, to a low concentration, which would be the left-hand side, but they're too large. In diffusion, molecules move from, same as osmosis, a high concentration to an area of low concentration. Okay? Next. What is homeostasis? The definition of homeostasis means same state. Cells get rid of waste products and take in nutrients. Kind of like in your house, you guys take out your garbage, you go to Publix or wherever you shop, and you bring nutrients in. So the whole point of a cell maintaining homeostasis is to keep a balance between too many waste products and enough nutrients. How does that happen? Well, one key thing is cellular transportation, active and passive transport. What do you know about passive transport? There's three things you should know. Passive, think about what it means. Does it require energy? No. What's the movement? Always from high to low. Definitely have to know that one for the exam. You should know passive transportation goes from a high concentration to a low. Great example that you should know after taking biology is osmosis. Water will always move from a high concentration to a low concentration. So that's passive transport. What about active transport? Well, when you look at active transport, it does require energy. It actually requires ATP. That's why if you see the word active in the transport part of it, that's what it's referring to, the use of energy. It's going to require energy because it's going to go from a low concentration to a high concentration. Movement of large molecules into cells is really what you're doing when you're doing active transport. All right, so next one, energy use in biochemical reactions. We know that in a cell, cells use ATP. So cells use ATP for energy. So there's a cycle between ATP and ADP. A phosphate breaks off. So in ATP, the T stands for tri, which means there's 3 phosphates. In ADP, the D stands for di, which means there's 2 phosphates. So the breaking and reforming of the bond is kind of like on your cell phone when you recharge your cell phone battery. So in this case, what you're doing is you're charging the molecule. ATP is a molecule that cells use for energy. And so when it has 3, it's like a charged battery on your cell phone. Think of it like this, you know, your little battery symbol. That's like a charged battery. ADP is when it only has a partial charge. Which cellular process produces ATP? That would be cellular respiration. Happens in the mitochondria. Should know that. The cellular process that produces ATP, that is definitely on the final exam. That's a keeper. Definitely know that. What do we use ATP for? Active transport, all biochemical activities of the cell. Everything the cell does. Putting molecules together, breaking molecules apart. All those things are done by using ATP for energy. What are the reactants and products for each of these? Well, photosynthesis. So, if you look at photosynthesis, the reactants for photosynthesis, what do you need for photosynthesis? You need water, you need carbon dioxide, and you need light from the sun. Those are the 3 key things you need for photosynthesis. The products are going to be glucose, C6, H12, O6, and O2. Because the byproducts of photosynthesis for plants are glucose, sugar, and O2. What cell does it occur in? Occurs in plant cells. Definitely need to know that. Well, if you take the photosynthesis equation, you flip it over, you get the cellular respiration equation. So the products of photosynthesis become the reactants for cellular respiration. So you need glucose plus oxygen, and what you're going to get on the other side is you're going to get water and carbon dioxide and ATP, because that's the whole point of taking the glucose is changing it into a form that's usable for plants, changing it into ATP. So if you look, plants and animal cells all do cellular respiration. It's aerobic, which means it requires oxygen. Cellular respiration, that's anaerobic. Typically happens in all cells, especially like bacteria and yeast. What you're going to have there is the reactant is going to be C6H12O6 glucose, but there ain't no oxygen. No oxygen, that's what makes it anaerobic, without oxygen. So what you're going to get instead is a byproduct is going to be CO2. You're going to get lactic acid. That's like when you work out and your muscles get sore. You produce a little lactic acid. The other thing you possibly could get is some ethyl alcohol, C2H5OH. You don't really need to know that for the exam. Key thing is you should know the reactants and products of photosynthesis, reactants and products of cellular respiration. Cellular respiration, anaerobic cellular respiration happens in all cells. Here's an example where you have to kind of break this down. If you look, I have photosynthesis right here. How do I know it's photosynthesis? When I look, I have water and carbon dioxide on the right-hand side. This is going to be cellular respiration. How do I know by looking at it? Well, I have glucose and oxygen. And finally, with just glucose on the left, this is going to be anaerobic cellular respiration. So this is going to be anaerobic respiration. Okay? So, what do you need to know? All right, so you have to be able to identify these reactions. Well, first question they ask you is which one's photosynthesis? Photosynthesis is going to be A, because water and carbon dioxide, just memorization, on the left-hand side, you're going to produce glucose and oxygen on the right-hand side. So the answer for 30 is going to be A. B, they want to know which one's fermentation. Anaerobic cellular respiration. That's only when you have one thing on the left-hand side. It's only going to be glucose. That's going to be C. Which of the reactions is cellular respiration? Remember, cellular respiration, aerobic. Aerobic means oxygen, so O2 has to be on the left. So bingo was his name. You have O2 right there. You also have C6H12O6, which you should know is glucose after taking biology. You're going to produce carbon dioxide and water. So the correct answer there is going to be B. Which reaction requires or stores energy? Which one requires or stores energy? That's going to be both A, B, and C. All require or store energy. A stores it. B requires energy for you to break the molecules apart, and so does C. They all involve ATP. Which reactions release energy or release ATP? That is going to be B and C. Cellular respiration releases ATP. In fact, probably should be added here. And anaerobic respiration releases ATP. Which reaction releases the most energy? That's going to be B. That's why we use cellular respiration, because it produces the most energy possible. Which reaction requires chlorophyll? Well, chlorophyll is what's in plants that makes them green. And what happens in plants? Photosynthesis. So the correct answer is going to be A. Which reaction requires light? Again, photosynthesis. Key component of photosynthesis is you need sunlight to make it go. So that's going to be A. Which organisms carry out process A? That's going to be plants. Plants photosynthesize. So those are going to be the ones that carry out process A. Which organisms carry out process B? All eukaryotes. So all eukaryotic organisms undergo cellular respiration. Next one. What is the function of enzymes in cells? Enzymes have one key function. They speed up chemical reactions. So the whole point, it's like a turbo boost for a car. Enzymes kind of speed up a chemical reaction. That's the definition of a catalyst. This is exactly the same thing. Enzyme is a biological catalyst. You definitely have to know that for the exam. It's definitely on there. What is the importance of shape to enzyme function? Enzymes are like lock and keys, like your house key. When you go home, you put your key in the door. It opens your door because it has a specific shape. Enzymes work on only specific molecules in the body. So in order to do that, they have a specific shape that allows them to work. Explain what determines the shape of an enzyme. Well, the key thing that makes the shape of an enzyme is the amino acid sequence. Because remember proteins, we learned about a macromolecule. All proteins are is a whole bunch of amino acids strung together. Well, depends on the kind of amino acids that you put together, makes the protein into a certain shape. So amino acid sequence is the primary factor in the shape of an enzyme. 43. Why can enzymes be reused over and over? They're not technically part of the reaction. All they do is speed it up. So they help break down or put together molecules, but they don't actually take part in the reaction. They're like little helpers. Since they're little helpers, they don't really get used up. How do extreme pH and temperature, like in changes in bodies and cells, affect enzymes? They cause them to denature. We talked about an enzyme having a specific shape, like your house key. What happens if you take your house key and you take a file to it and you saw off some of the teeth of your house key? It won't have the same shape. Once it doesn't have the same shape, it's not going to work anymore. So extreme changes in pH or temperature cause enzymes to lose their shape. That's the word denature. So once an enzyme loses its shape, kind of like your key at home, it won't fit in the door, it won't open the door, you got a problem. Label the picture of the following enzymatic reaction. If you look, you have to be able to see this. The enzyme, notice, isn't changed. So that I know this, the bottom part is going to be the enzyme. So that's going to be the enzyme. Substrate is the molecule that the enzyme works on. So it's going to take this molecule. This molecule will be the substrate. Then the product at the end is what's left. Enzymes only do two things for your body. They either are scissors, they break apart molecules, or they're glue sticks and they stick them together. So if you look at this diagram, the enzyme doesn't change. There's the enzyme. This is the substrate going in, fits in here. The enzyme substrate complex is when they're just connected together. That's this right here. It's going to be that. And then finally, the product is either two pieces, because an enzyme's taken one molecule, broken it apart, or is you take two molecules and you put it together. That's the whole point of enzymes. Enzymes, most of the time, end in ASE. That's how you spot them. So an enzyme, how do you spot an enzyme? It has ASE. For example, lactase. Lactase helps you to digest lactose, which is in milk. People are lactose intolerant because they're missing the enzyme lactase. Amylase is another enzyme. It ends in ASE. It's one of the ones in digestion. It helps break down carbohydrates when you eat something. In your saliva, you have amylase, which helps break it down. What temperature is the best for this enzyme to work? When you're reading an enzyme graph, you're looking, the best temperature is the top, the peak. So in this area right here, the top of the peak is the best temperature. So if I drop down there, I would say that's right around 37. What do you look for? You look for the top of the peak. That will be the best temperature that enzymes work at. Which number, Roman numeral 1, 2, 3, or 4, does it start to denature? Denaturing means that it loses its shape. So when it starts to denature, what happens is you'll see enzyme activity start to drop. See the little red highlighted area there, how it starts to drop? So the denaturing process actually occurs in the Roman numeral 3 section. What do you look for? You look for a drop in enzyme activity. That's how you spot it. So you look for a drop in enzyme activity. Next one. What is the pH best for enzyme X? Well, here's enzyme X right here. Well, best way to figure it out is take the top of the curve, drop down somewhere between 3 and 4, we'll say 3.5. What about what pH is best for enzyme Y? Look at the curve for enzyme Y. If you look, you drop it straight down, sort of, that's not exactly straight down, probably between 9 and 10. The trick to reading enzyme graphs, though, maximum activity occurs at the top of the peak. So find the top of the peak in the graph, and then all you do is you trace it all the way down. Piece of cake. All right, we're going to stop there. This is the end of episode 1. And click on the episode 2 and episode 3 to finish the rest of the exam review.